Okay, good afternoon, everyone. Today, am I audible? I'm audible, right?
Yes, sir.
Everything is
Yes.
Yes.
okay. So, from today and in afternoon, we will be discussing with RNA structures. So, by the end of this series of lecture, you will be able to appreciate complexity of RNA molecules than you might have thought about before. Okay, so RNA are way too much complex molecule. Okay, so far their structure is concerned, and these lectures uh, are supposed to be the one that would increase your understanding about the structures of RNA. Okay, so let us begin. This is what we are going to do in the entire day today. First, I will let you know how RNA differ from DNA and what is that make RNA so special. Okay, so I won't go in more details of the basic structure of RNA. Why? Because since it is a nucleic acid and the lectures on DNA structures are already done by Desai sir, right? DNA के lecture हो गए ना?
Yes, sir.
Yes.
Yes.
So you are already aware of the structure of nucleic acid. The basic has been already done. So now I would only focus on stuff like how RNA differs from DNA and what are the special properties of this RNA that makes it so different from DNA. Okay. Then we would discuss the structure of RNA. Just like as we have discussed the structure of protein. First, I would let you know about the primary structure, then we would discuss secondary structure and tertiary structure of RNA. Okay. The next topic that we will end up will be after lunch, in which we would begin with the discussion of representation of RNA structures. What are the different methods by which you can visualize your RNA molecule all along the primary, secondary, and tertiary structure. Okay, what are the what are various representation methods that are developed for visualizing the RNA data? That is what we are going to deal with. I mean, deal with first when we begin our next session after lunch. Okay, and finally, I would end my discussion with energetics that is involved in RNA structure. So, in short, these are the four topics that we are going to cover in today's four lectures as such, or today's 4 hours as such. Okay. So now let us first discuss how RNA differ from DNA and what makes RNA so special. Okay. So just to begin with, as you know, in case of protein, one sequence usually leads to a unique structure, and this unique structure usually perform unique function. Okay, so our objective is that we shall focus on how similar stuff happens in RNA as well. Okay, how RNA sequence helps it to get one unique structure, and how one unique structure might help it to gain unique function. Okay, so this is what we are going to talk about. You know, the same thing that is applicable for protein is also applicable for your RNA as well. Okay, so sequence will give rise to structure, which in turn will give rise to a unique function. Okay, so now we'll say, for example, some RNA molecules are relatively unstructured, right? So they can be elongated in nature, some parts of mRNA, while on the other hand, some RNA molecule can have very intricate, elaborate, and very complicated structures. Well, say for example, in case of tRNA, in case of ribozymes, even ribosome and riboswitches, these are all, majorly made up of RNA molecules. Okay, so what you see over here is the structure of a typical ribose, which we are going to deal with in a little while. Okay, so this ribose switch structure was, published in Nature Structural Biology back in 2012. Okay, so this is where I have taken this picture from. Okay, so these RNA molecules are very often highly complex in structure. Okay, and somehow these RNA molecules are involved in all aspects of gene expression, right from synthesis of DNA to replication to transcription to translation. Somehow these RNA molecules are involved. Well, this indicates that it has been an integral portion, or it has been an integral or very crucial element that has been maintained right from the beginning of life. Okay, so here is the structure of your ribosome. Okay, so a few examples of its very intricate functionality involves, like, you can consider these G ribose switches. Okay, so these G ribose switches are RNA molecules that act as natural biosensors. Okay, and they function usually in bacteria for sensing if there are metabolites in the environment and signal back to cells, so that the cell can respond to the surrounding metabolites if there are any. Okay, so the first structure of the ribose switches were published in 2004 by Bete et al., which was published in journal this Nature. Okay, so I would advise you to read more about how these RNA act as natural biosensors in form of these G ribose switches as such. Okay, so some of the RNAs are catalytic RNAs. Well, say for example, your self splicing group 2 intron. Okay, so this is how your self splicing group 2 introns look like. So cell uses the catalytic activity of this self splicing group 2 intron to cut and stitch RNA molecules for gaining functional stability. Okay, not all RNA who has got functional nature are created as they are. Okay, so in order to gain the functional stability, some of the portion of that RNA molecule needs to be cleaved off, so that it would become mature and it can use itself for, for gaining the functional stability. Okay, as everyone knows that protein synthesis factories, what we call them as ribosomes, are probably one of the most largest and highly structured RNA molecule that we have studied it. Okay, so the structure of intact RNA was published by Venki Ramakrishnan back in Nature in 2009. This is the structure of bacterial ribosome, for which he also received the Nobel Prize. Okay, so as you can see over here, you have got a tRNA, you have got 50S subunit, you have got 30S subunit. The black colored portion that you see over here is mRNA, right? And it is the protein that is being synthesized. So most of the portion of your ribosome is composed of RNA with a few proteins that are flanked nearby to provide the stability. So the core catalytic portion where the protein synthesis usually occur is the RNA portion. Okay, so it is your RNA that is responsible for performing the catalytic function of joining two amino acid residues during the protein translational process on the ribosome. Okay, this mRNA, which I have told you, it can be unstructured, it can be present it can be in an elongated form, because the thing is that whenever right from your 11th or 12th and during graduation, whenever people or teachers usually talk about mRNA, they usually draw such a plain line indicating that this might be a straight RNA sequence. Okay, so it is not like that. mRNA also contains some highly structured regions, and as you can see over here, these highly structured regions that are present in your mRNA, those are present on the 5 prime and 3 prime UTR, what we call it as untranslated region, that are usually flanking to the coding sequence from which your protein is supposed to be synthesized. Okay, and these 5 prime UTR and 3 prime UTRs that are present or that are shown over here, well, they are known to play a crucial role in regulation of protein synthesis. Okay, so it is not like your mRNA is always a straight strand of nucleic acid that is usually represented on board. Okay, this mRNA also contains some highly structured region in its 3 prime and 5 prime end, which we call them as 5 prime UTR and 3 prime UTRs as such. Okay, so now let us talk about the chemical structure of RNA. Okay, so as you know that of course this is going to be a nucleic acid. That means it would contain a backbone, and this backbone would be attached to a base, right? So or what we call it as purine or pyrimidine base as such. Okay, so the backbone is usually formed of phosphate and a sugar moiety. Okay, and this backbone, phosphate sugar, phosphate sugar, phosphate sugar, this backbone moiety would be attached to one of the bases as such. Okay, so in case of RNA, in place of T or thymidine, you have got uracil. All the remaining other 3 bases are same in DNA as well as in RNA. You have got G, you have got A, you have got C, but in place of thymidine, you have got uracil. And your uracil is nothing but thymidine that lacks a methyl group at the 5th position. Okay, do you see the structure of uracil over here? 1, 2, 3, 4, and 5th position, it don't contain methyl group over here. Or you can just say your uracil is thymidine that miss one methyl group. Okay, so here is where the primary structure of DNA differs from RNA. Okay, this is one difference, and another important difference is the presence of hydroxyl group at 2 prime position in your sugar molecule. Okay, so this is 2 prime position. It contains a hydroxyl group over here, which differs in case of RNA, which differs in case of DNA. DNA don't have this 2 prime hydroxyl group. Okay, and RNA has got its own 2 prime hydroxyl group. Okay, so here is the basic difference. Okay, now coming to this OH group, it is this 2 prime OH group that endows RNA its special power. It is what gives RNA, I would say, superpower, just like Wolverine, which enables it to do some extra functions which your DNA cannot perform. Okay, so now let us talk about what superpower this RNA has, and the superpower to this RNA is given by this 2 prime over 2 prime OH group, and how this superpower is endowed on your RNA molecule. Okay, well, say for example, if you consider the chemical structure of the sugar group, what you can see that you have got this hydroxyl group, and the hybridization state of this oxygen is in such a way that it has got a lone pair of electron, and due to this presence of this lone pair of electron, it can act as acceptor. And since this OH group has got hydroxyl group which contains hydrogen, therefore it can also act as hydrogen bond donor as well. So in this way, it can make hydrogen bond in the capacity of acceptor as well as in the capacity of donor. Okay. So all in all, what I want to say is that this 2' OH can accept as well as donate hydrogen bonds. Okay, so in this way, every 2' OH can possibly have 2 hydrogen bonds. Okay, and since it can have 2 hydrogen bonds, it makes your RNA backbone sticky. It can somehow attach to different substrates. Okay, and this particular sticky nature, which is endowed by the hydroxyl group, enables your RNA to make numerous interaction that DNA cannot make. DNA में hydroxyl group नहीं होता है, therefore it is not sticky, and hence it cannot interact with various chemical groups that are present in its vicinity. Okay. So when people talk about this nucleic acid structure, well, most of the time people think that it is the sequence of bases in DNA and RNA molecules and bonding between them that makes these molecules special. Okay, but you should also know that a lot of things are going with sugar and phosphate backbone as well. Okay, so the structure of sugar, it is the structure of your sugar that gives DNA and RNA their distinctive shapes and structures as such. Okay, so it is not the only basis that makes your RNA molecule so special. Okay, a lot more things happens over sugar and phosphate backbone as well in RNA. Okay, so first thing you should remember is that sugars are not flat. Sugar molecule कभी flat नहीं होता है. Okay, so the ribose in sugar, okay, ribose sugar in your RNA are usually 5-membered ring. Okay, of which the 4 carbon atoms are present inside the ring, while the 5th carbon atom is a part of the backbone, which is attached to your phosphate group as such. Okay, so now you should also remember ki, the nomenclature of sugar moieties. So far we are concerning your nucleic acid structures. The nomenclature of the carbon nomenclature in RNA is like they have given numbers like 1', 2', 3', 4', and 5'. Okay, so what might be the need of giving this prime in naming the sugar molecule? Have you wondered about it? Any suggestions on this?
To differentiate them from the carbons of nitrogenous base.
Yes, exactly, because there are also carbons that are present in nitrogenous bases. So that there should be no confusion when people talk about carbon 1 or carbon 2. We always, when we talk about like carbon number 1, carbon number 2, and we are talking about nucleic acid, that would mean that we are talking about the carbons that are present on the bases. While if we are talking about like carbon 1', 2', 3', and 4', that means we are talking about the carbons that are present in your sugar backbone. Okay, so these carbon are supposed to be buckled or puckered out of plane. Okay, so since these are not flat, that means these often one of the atoms of one of the carbon atoms from your sugar group will kick its knee out of its plane. Okay, and what kind of carbon atom kicks its knee out of its plane? We have two type of puckering. Okay, what we call it as a 3 prime endo puckering and 2 prime endo puckering. Okay, so in case of this puckering यानी क्या? One of the carbon atoms from the sugar would leave its plane, and hence we say that we have got carbon puckering happening over there in RNA molecule. Okay, so there are two major conformation of sugar pucker, what we call it as C3 prime endo sugar pucker, in which the carbon at position 3 prime will get out of the plane of the sugar molecule. Okay, and this is most common sugar pucker that usually happens in RNA. Okay, now there is one interesting fact that since it is the 3 prime carbon atom that is puckered out of the plane in case of RNA molecule, and it is the 3 prime carbon atom that is associated with the phosphate group. Okay, therefore now if the 3 prime sugar atom, sorry, 3 prime carbon atom from the sugar group is displaced out of plane, that would mean that the phosphate group that is associated with this 3 prime carbon atom will eventually also get which eventually also get pulled along with the 3 prime carbon atom, and due to eventual pulling of this phosphate group all along with the 3 prime carbon atom, the distance between the phosphate along the two nucleotide is smaller in case of RNA. Or what I can say is the interphosphate distance between the two nucleotides in your RNA molecule is smaller. It is about 5.9 angstrom in an RNA helix, while in case of your DNA molecule, it is the 2 prime atom of 2 prime carbon atom from your sugar that moves out of the plane. Okay, and since this 2 prime carbon atom is not associated with sugar, therefore the interphosphate distance in case of DNA is longer. That is, it is 7.0 angstrom in an helix. Okay, so it is specific puckering of carbon atoms in this C3 prime endo group, okay, or this C3 prime endo puckering is responsible for differential interphosphate distance, so that structure of RNA differs from structures of DNA, and it has got a far-reaching effects in structure of RNA, which we are going to eventually discuss later. Okay, so the differences in the interphosphate distance in RNA as compared to DNA is due to the 3 prime puckering that happens in RNA, which don't happen in DNA. In DNA, there is 2 prime endo sugar pucker. Okay, and that eventually affects the interphosphate distance. Okay, so this is what we are talking about. एक और चीज आपको पता रहनी चाहिए कि ये जो पकरिंग हमेशा मतलब रेफर होता है, well, this puckering is usually referred with reference to the plane of the base. Okay, it is this 3 prime sugar 3 prime carbon atom that is pushed towards the plane of the base. Okay, in case of RNA, and it is the 2 prime carbon atom from your DNA molecule that will be pushed towards the plane of your base. Okay, so in this way there is difference in interphosphate distance in your RNA and DNA. Okay, so now what might be the biological impact of these differences in interphosphate distance? Okay, So first and foremost would be, due to differences in interphosphate distance, these DNA duplexes are elongated and spread out as compared to your RNA duplex. Okay, and this is very important so far the helical shaping of DNA and RNA are concerned. Okay, so when we say that these DNA duplexes are elongated, that means, well, say for example, if there are 10 bases or 10 nucleotide in DNA, they would require you can say they would require much space as compared to the 10 bases if they are present in your RNA molecule. Okay, your RNA molecule will stack those 10 bases in a smaller space as compared to your DNA molecule, which would require longer space. Why? Because the DNA molecule are elongated and spread out as compared to RNA molecule. Okay, so now uh, that we have covered some very important aspect of primary structure of RNA, we shall begin discussing its secondary structure. तो अब तक जो कुछ भी अपन ने discuss किया, we were discussing what? We were discussing the primary structure of your RNA molecule. Okay, and here onward we would learn some important terminologies. Well, say for example, RNA secondary structures and how they are formed by base pairing and so on and so forth. Okay, so to begin, well, let us talk about RNA secondary structure. Okay, so in case of protein, you have got very, intricate secondary structures available. Well, say for example, you have got alpha helices, you have got beta strands, which eventually collect up to form beta sheets. Then you have got turns, and then you have got coils. Okay, but you don't have got such a variety of secondary structures in RNA. Okay, so this RNA secondary structure is usually formed by RNA base pair. Okay, so what you see over here is a simplified representation of the most common secondary structures that are found in RNA. Okay, so what you see over here is a two stem capped by RNA loop, two RNA stems that are capped by hairpin loops as such. Okay, so over here, what you see, we call it as stems. There are two duplex stems that you see over here, and these two duplex stems are formed by base pairing, as you can visualize over here. Okay, what you have over here is something called as the junction, and these junctions are substructures that join the two stems. Okay, and above what you can see over here are called as hairpin loops. Okay, so these hairpin loops are for capping the stems. Okay, and these hairpin loops can be of tri or tetra form. Okay, depending on how many nucleotides are present in your loop. So if there are 4 nucleotides present in your loop, you would call it as tetra loop. If there are 3 nucleotides present in your hairpin loop, you would call it as tri loop as such. Okay, so this is the very basic secondary structure of your RNA molecule. Okay, now let us talk about how base pairing happens in your RNA secondary structure. Okay, so this normally A would pair with U and G would pair with C. But along with this normal base pairing, you also have alternative base pairing wherein your G would base pair with U, and this is what we call it as the Wobble pair. Okay, so it is not only the Wobble pair that is unusual. Okay, there are also other type of unusual base pairing which we are going to deal with in a little time. But let me first give you some details of the structural aspect of this Hubble base pair, and let us compare it with the normal AU base pair. Okay, so in normal AU base pair, okay, the atoms that are involved in hydrogen bonding between the bases are that of, well, say for example, the atoms at position 3 and 4 are involved in normal AU base pairing. ओके, जो आपका जो बेस का रिंग होता है उसमें से which atoms are involved in hydrogen bonding so far normal AU base pairing is concerned? The atoms that are placed at position 3 and 4 are involved in base pairing. On the other hand, in case of Hubble base pairing, in which your G is paired with U, the line of hydrogen bonding is somewhat different. Okay, in this case, atoms at position 2 and 3 are involved in base pairing, in contrast to 3 and 4 in case of normal base pairing. Okay, so now due to different hydrogen bonding arrangement, okay, the atoms at position 2 and 3 are involved in base pairing in case of Hubble base pairing as such. Okay, so now due to this shifting of hydrogen bonding lining, this uridine would be displaced upward in Hubble base pairing. ओके, ये इसकी वजह से क्या होगा, ये लाइन चेंज होने की वजह से आपका जो यूरिडीन है वो नॉर्मल बेस पेयरिंग के कंपैरिजन में इट वुड बी डिस्प्लेस्ड ऑन द upward direction. Okay, it would be displaced in upward direction, and eventually this would result in projecting the amine group, okay, amine group into the minor group. Although this amine group is now entered in the minor group, it do not create any kind of disturbances or it creates no perturbation in structure, and therefore it has the same stability as the normal AU base pairing. Okay, and hence such Hubble pairing is readily tolerated in RNA secondary structures. Okay, so although the lining of hydrogen bonding is different in your Hubble base pairing, but it won't eventually affect the secondary structure of your RNA molecule. Okay, so these figures are taken from Watson, the book of Watson, if you have any. Okay, so this is what I was talking about, that it is not only this Gu-Hubble pair that is unusual in RNA. There are many other weird type of RNA base pairing that people have discovered. Okay, so there is something called as AC plus Hubble pair, in which A can also pair with C, but this can only happens when the cytosines become protonated at certain pH. Okay, people have also documented that U can also base pair with U, uridine can also base pair with another uridine. Sometimes this G is also found to be base pairing with A. Okay, now it is to be noted that both of these G and A are purines. That means they are both double ring. Okay, and hence they are larger in size. And if GA pairing is happening in your RNA secondary structure, that usually happen at the end of the duplexes where it requires more stability. Okay, and hence this GA base pairing is usually seen in the RNA that is usually associated with the stabilizing effect at the ends of the duplexes as such. Okay, so these are all alternative base pairs that people have documented and published in the literature. Okay, so now let us talk about other secondary structures that are present. RNAs are also associated with bulges and loops. Okay, Now these bulges and loops, as you can see over here, this is called as a loop while this is called as a bulge. Okay, so these bulges and loops, well, they play a very important role in stabilizing the overall structure of your RNA molecule. Okay, so these bulges, well, they can be either symmetric or asymmetric. This is over here, what you can see is a symmetric bulge. Why? Because it happens only on one strand. Over here, नीचे जो दिख रहा है आपको, this is called as symmetric bulge. Why? Because the bulge is present on both the strands as such. Okay, so this asymmetric bulge, if there is any, that can result in bending of the duplex. Okay, तो ये asymmetric bulges से क्या होता है, आपके duplex bend हो सकते हैं. Okay, while if there are symmetric bulges, well, that would eventually affect the flexibility of RNA molecule. Okay, so due to presence of these symmetric bulges, mobility around these symmetric bulges somehow would be affected. Okay, so you can read this paper that was published in Journal of Molecular Biology back in 1996. It is very classic paper that demonstrate what is the effect of bulges and loops in RNA. Okay, and these figures were taken from Watson book. Okay, so I would advise you that you read this paper. It is very interesting one. Okay, so now yes, this is one more thing that I want to tell you guys, that uh, in most of the books you would see that when people talk about these bulges, symmetric or asymmetric, they show something like this. Okay, so in real RNA structures, well, the bases do not stick out of uh, out of your strands. Okay, out of your secondary structure. Okay, these nucleotide bases have aromatic properties, and hence they have got tendency to avoid contact with water. And what do these do is that they stack upon each other to avoid contact with water. Okay, and therefore what you can see over here, that in this adjacent molecule you have got two unpaired bases. Okay, see how beautifully. However, you have got unpaired bases, but they tend to avoid the contact with water by stacking themselves one upon each other. Okay, you can download this particular RNA molecule which has got PDB-ID 255D. Okay, and you can see that there are two unpaired bases in this structure. Most probably they might be present over here or over here. Okay, so what I want to say is that do not have an impression that the bases will stick out of your secondary structure when at cellular conditions. It don't happen such way. Okay. Okay. Now let us talk about in the conservation in the RNA hairpin loops. Okay. So now these RNA hairpin loop, which are present on the termini of the duplexes, are usually highly conserved in sequences as well as in their structures. Okay, they usually have UUGC or GAA, what we call it as tetra loops. Okay, so there are many structures with UUGC as a terminal loop, and their three-dimensional structure comparison revealed the fact that they have similar 3D structure. Okay, so over here also, the similar sequence would fold up into similar structure as in analogous to the protein structure. Okay, so this particular terminal loop which has got this UUCG as a hairpin loop, you can download it from PDB using this PDB identifier to KOC. Okay, so all in all, what I want to say is that if there are hairpin loops, they can have conserved structure as well as they can have conserved function. Okay, good. Now, well, let us talk about some RNA secondary structure, how RNA helices actually look like. Okay, so your A form of RNA, which is highly regular, okay, it has got, of course, minor groove and major groove. Okay, this minor groove in RNA is large and flat as opposed to DNA. In DNA, it is the major groove that is large and flat. Okay, the major groove is narrow and deep in RNA, which is opposite in DNA. Okay, in DNA, it is the minor groove that is narrow and deep. Okay, so it is the secondary structure or the helix of an RNA is more compressed as compared to that of DNA. Okay,
Hello, sir.
हाँ बोलो.
Sir, if it is vice versa of DNA, then why it is called as minor and major groove?
Okay, so it is just for the sake of comparison purpose. Okay, so just to give you a perspective of how it differs from DNA. Okay, so it is all up to us to call it what kind of groove it is, right?
Okay, sir.
Okay, ठीक है. Good. So now it is, not just the atoms that are present in its primary structure that would shape the RNA secondary structure. Okay, so waters are also known to play a imperative or very crucial role in shaping the RNA secondary structures. Okay, well, most of the story begins back in mid 90s, when people started to crystallize these RNA molecules with very higher resolutions. Okay, so what these people observed is that water play a very important role in shaping the structure of nucleic acids. Okay, now when in mid 90s, as you can see that this paper were published in 1995 and 1996. At that time, these two group were able to crystallize very ultra high resolution models of RNA. And what these people observed is that these waters are an integral part of your RNA structures. Okay, so what happens is that whatever shown over here, जो दिख रहा है आपको यहाँ पर रेड कलर में जो दिख रहे हैं, well, these waters, well, they form regular lattices that connects the polar group in your RNA molecule. Okay, so it is not only the atoms that are present in the sugar phosphate backbone as well as bases that will give its secondary structure, but the surrounding waters also play an important role in stabilizing the RNA secondary structures. Okay, so you might read this paper to gain more insights into that. Okay, so this is the basic comparison. Okay, तो देखो, this is B form of DNA, A form of DNA. Well, this slide is quite self-explanatory. Okay, so I would advise you to pause on this slide when you study. Okay, you might kind of expect questions from this slide as well. Okay, so remember that due to interphosphate distance in DNA and RNA, differences in the interphosphate distance, the B form of DNA is more elongated as compared to A form of RNA. Okay, and you can compare the other properties as well. Okay, so now let us move on our discussion to some more details, like how are RNA secondary structures formed. ओके, अपन काय के बारे में बात करेंगे अपन की अभी वेल कि exactly what are the factors that affects the formation of RNA secondary structures. Okay, so well, you can read this review which was published in annual review of plant biology that was published back in 2016. Well, this is an excellent review on RNA secondary structure, which gives you a kind of insight on what are the forces that affects the formation of RNA secondary structures. Okay, so there are at least four major forces or four major factors that affects the formation of RNA secondary structure. We are going to discuss all of these four factors one after another. Okay, and these four factors being transcription, steric crowding, RNA binding proteins, or what we call it as chaperone proteins, and what are the interacting ions that are present in its vicinity. So these are the factors, you know, that affects the formation of RNA secondary structure as well as maintaining the secondary structures of your RNA molecules. Okay, well, now how transcription might be one of the factors that affects the formation of secondary structure. Okay, so the thing is that RNA folding is supposed to be co-transcriptional. Okay, so that means the moment RNA is being synthesized, it start to it has got sequential folding. Okay, and since it is a co-transcriptional phenomenon, the formation of RNA secondary structure thus can vary with the speed of RNA polymerase elongation would be different for different RNA polymerases, right? So depending on the speed of RNA polymerase elongation, the formation of RNA secondary structures can also vary. Okay. Second most factor that affect the formation of RNA secondary structure is that what kind of proteins and ribozymes are in vicinity of your RNA molecule. Okay, so it is said that RNA folding is guided by proteins and ribozymes. Okay, so these proteins like RNA chaperone have got RNA chaperone activity, and this RNA chaperone activity controls initial RNA secondary structure formation. Okay, so due to the RNA chaperone activity of these protein, well, what do these RNA chaperone proteins do is that they kind of, you know, enable that RNA molecule to avoid the kinetic folding traps, or what we call it as local free energy minima. Okay, so that due to their help, RNA can bypass these local free energy minima and attain the lowest free energy states. Okay, and it is this RNA chaperone activity that help them to avoid improper gaining improper conformations as such. Okay, so these proteins also play very important role in shaping the structure, or you can say shaping the secondary structures of RNA. Okay, well, now let us talk about these RNA chaperones. Okay, these RNA chaperones constitute a very diverse group of protein. Okay, and these group of protein are functionally defined through their ability to facilitate RNA folding. Okay, and the proteins which usually contain this dead box helicase domain are known to facilitate RNA folding. Okay, now this RNA
Hello,
folding,
sir.
bol beta.
Will you explain the dead box helicase domain?
Uh, well, it is, you know, a specific three-dimensional tertiary structure that are present in the structures of those proteins. Okay, you can say that just like immunoglobulin domain, okay, these are specific domains that are present in, say, some proteins that facilitates that protein to recruit RNA and help them folding. Okay, well, let us not go in more details of what secondary structures are there and how do it. But for a time being, it is sufficient for you to know that if a protein contains dead box helicase domain, that can be an essential marker that you can consider whether it would go and bind to RNA or not.
Sir, why it's called as dead? Is it DEAD are the amino acids?
No. Specifically, I don't remember this thing now. Okay, you can read this paper if that is the case. Okay, I will let you know about this thing. Okay, if this DEAD is an amino acid residue or it has got some different meaning. But for time being, I don't know. Okay.
Okay, sir.
Okay. So this RNA folding or RNA refolding can be either ATP dependent or it can be ATP independent. Okay, it might utilize energy to refold it, or it can be independent as well. Okay, good. We are going good. We are going good. So now these RNA chaperones are known to stabilize RNA folding intermediate, and that would enable a complete conformational search. Okay, so these chaperones generally lack sequence specificity, and due to this, they might have a wide array of potential target. ओके तो ये आरएनए शेफरॉन प्रोटीन्स जो होते हैं वो आरएनए का सीक्वेंस आइडेंटिफाई नहीं करते. On the other hand, they identify the structure of RNA if it is misfolded. Okay, and in this way, these proteins, these chaperone proteins, are able to have multiple targets that they can identify and refold properly. Okay, so now if there is a loss of RNA chaperone activity in any cells, well, that is known to result in widespread misfolding and altered phenotypes in plant. Well, so for example, if RNA chaperone activity is lost of, say, some protein, that result in osmotic stress and even cold shock. Okay, so it is an important function that is necessarily be able to maintain to sustain life. Okay, then you have got these RNA binding proteins. Okay, so these RNA binding proteins contain RNA binding domains. Okay, these RNA binding domains would preferentially bind to specific structural conformations, okay, of RNA, and depending on what kind of structural conformation these protein bind, you can have, like, there are some protein like they have got RNA recognition motif and K homology domain. Well, they bind to single stranded RNA. Okay, while uh, if you have this double stranded RNA binding domain in, say, some protein, they are known to bind the double stranded RNA. Okay, it completely depends on the structure of your protein, whether they would recognize the single stranded RNA or whether they would recognize the double stranded RNA. Okay, so these RNA binding proteins can also target specific structural patterns. Well, say for example, if the protein has this sterile alpha motif domain, it can specifically target the stem loops. Okay, and this RNA and RNA binding proteins are known to interact in induced feed fashion. That would eventually mean that when they come close to each other, there would be conformational changes in both of them in order to perform their function. Okay, so this is what it is. And the final factor that affect the formation of RNA secondary structure is the presence of ions. Okay, so now it is known that these monovalent cations like K plus ions is the most dominant species in cell, and these K plus ions well, are the one that are known to regulate the formation of RNA secondary structures. So now you know that RNase phosphate will impart the negative charge on the RNA structures as such, and this K plus has got positive charge, and your phosphate and eventually RNA has got negative charge. Therefore, this cation phosphate interaction would bring about short range ionic interaction. Okay, and due to this short range ionic interactions, hydrogen bonding becomes an easy way to happen, and it also enable base pairing to stack upon each other. Okay, it is these K plus ion that are said to guide stacking of bases upon each other properly. Okay, so these cations and phosphate from RNA are known to stabilize the compact secondary structures. Okay, so if this is the primary structure of the RNA, it is the monovalent cations that impart the initial secondary structures. Okay, so मैं ये सारी चीजें इसलिए बता रहा हूँ because well, I want you to appreciate that there are a lot of factors that somehow define the secondary structures of RNA. Okay, कौन से factors देखे अपन ने? We have considered factors like there might be ions that might affect the formation of secondary structure. There are different type of proteins, okay, that might affect the formation of secondary structures. There are transcriptional events, okay, and there might be steric crowding as well. Okay, तो ये जो 4 factors है, these are the external factors that are present in vivo that affects the formation of secondary structures, right? Do everyone agree with this?
Yes, sir.
Okay, so now the correct in vivo structure of RNA may and eventually differ from the in vitro or in silico prediction that you might make. Okay, so there are algorithms, okay, that might take up your RNA sequence and give you a kind of folded RNA structure, right? But it is not 100% guaranteed that the predicted RNA structure would be the same that would happen in your cell. Okay, or even if you try to fold your RNA in test tube, right? It might not be the same one that might happen in cells. Why? Because at in vitro condition or in silico condition, you are not considering the factors that are that we have discussed for time being, right? वो 4 factors आप in silico prediction में consider नहीं करते, या फिर in vitro prediction in vitro folding में consider नहीं करते, and therefore the in vivo structure of RNA may be different as compared to what you see in vitro or what you predict in silico. Okay, तो ये सारी झंझट मैंने किस लिए की? Just to let you know कि okay, there are lot more factors that affect the structure of your RNA, and what you will predict in your next semester might not be the reality. Okay, why? Because there are other factors as well that might shape the RNA structures. Okay, so keep that in mind as well. Okay, so this is what I have for time being. Okay, we would discuss the remaining portion after the lunch session. Okay.
Okay, sir.
Okay.
ठीक है.